Today we are going to discuss about promoters, um, about their roles and what are the difference in promoters of prokaryotes and eukaryotes. So I'm Dr. Rajiv Basumatari from DR College Golaghat. Okay, let's start. So promoter is a specific location in gene where the enzyme RNA polymerase can bind or sit before transcribing the gene for the synthesis of protein. So for protein synthesis, we all know that there are main two processes, that is transcription and translation. So during the process of transcription, the DNA sequence or the sequence which is in the gene is first copied as a mRNA molecule, and in the next step from this mRNA, the amino acids are translated or the polypeptide chain is formed, which later on forms into protein. So for the process of protein synthesis, transcription and translation are the main processes, and during transcription, what happens, the copying of DNA to mRNA is done by an enzyme called RNA polymerase. So RNA polymerase, it will work in the DNA molecule or in the gene segment, or in the gene, it will work, and then it will start copying the DNA sequence into mRNA sequence. Let us understand how the promoter helps in the process of transcription. Let's assume this part of DNA is a gene, and just before gene there is a sequence which is actually identified by enzyme RNA polymerase, and the RNA polymerase can identify that sequence, and they can bind with this portion of DNA, and this portion of DNA is known as promoter, so which is our topic today. So after binding with the promoter, RNA polymerase, it will move in the gene, and it will start copying a DNA strand into mRNA. So the strand from which the mRNA is copied will be called as template DNA, and the another strand, the opposite strand of the template, is known as coding strand, template DNA strand. The part or the exact location from which the copying of mRNA will start, for example, from this point, the mRNA will be copied. So this point is actually the transcription start point, and from this point the template DNA sequence will copy as mRNA sequence. This is 5' and 3' in this side, and this bases are numbered, actually they can be numbered. So this base at the transcription start point is numbered as +1, and the bases on that side, on the side of the gene, they are counted as positive. So for example, the base at the start point is +1, then next base pair will be +2, then +3, and so on. Okay, so the bases which are located on this side are known as downstream base, and on that side, on the left side of the transcription start point, they are numbered as -1, -2, -3, and so on. So left side is numbered in minus and the right side in plus. So the base on the left side, or the elements which are located on the left side, are known as upstream. Okay, so you have to remember these two terms. There is downstream, that is towards the right side of the transcription start point, and towards the left, they are known as the upstream bases or upstream base pairs, so you can say like that. So what happens, the promoters The promoters for prokaryote means those bacteria, promoters in the genes of the bacteria. These promoters are exclusively located at the upstream on the left, that means on the left side of the transcription start point. All the promoters of prokaryotes are upstream promoter, means they are located on the left side of the transcription start point. But in eukaryotes, or in the RNA polymerase, there are three types of RNA polymerase enzymes in eukaryotes: RNA polymerase 1, 2 and 3. And the promoter for RNA polymerase 1, 2 and 3, they may be located, some parts are located at the upstream side, and there are some promoter regions which are located at the downstream. So this is the main difference in the location, about the location of the promoter in prokaryotes and eukaryotes. While understanding promoter, we have to understand one more concept that is known as consensus sequence. So what is consensus sequence? So consensus sequence is a sequence of DNA within the promoter, consensus sequence of promoter. So consensus sequence of promoter is a sequence within the promoter which is constant in terms of their base sequence as well as in their function. Function means the function of the consensus sequence is to bind with the RNA polymerase enzyme. So the point is that the promoter is a long sequence of DNA, and within the promoter all the bases which are present, all the base sequence are not actually important for RNA polymerase to identify the region. So within the promoter region there are some areas, there are some specific areas actually which are identified by RNA polymerase, and that is why all the promoter segment is not important for identification by the RNA polymerase enzyme. Only the sequence, which we call as the consensus sequence within the promoter, only the consensus sequence within the promoter is important for identification by the RNA polymerase enzyme. This consensus sequence within the promoter is always constant in all the genes, all the promoters of all the genes. You see, there is a technical point. It is like that. There is only one type of RNA polymerase enzyme. So RNA polymerase enzyme is only one. Okay. For example, in bacteria there may be many genes. There may be 100 genes. There may be 500 genes. But there is only one type of RNA polymerase enzyme. And for all those 500 different types of genes, this only one RNA polymerase has to carry on the process of transcription. So that is why how only a single type of RNA polymerase can identify different types of genes or the different types of promoters of different genes. And it is also found in the study that the promoters of different genes are not equal in their sequence. So when you compare the promoters of different genes, you will find that the sequence of this promoter of a gene is different from promoter of another gene. Then how a single type of RNA polymerase can identify different sequences in promoter? So in a study, scientists have found that within the promoter actually there are some regions which are found in all the promoters, and which region or which sequence is constant in all the promoters. The other area is not similar in all promoter, but there are some sequences or there are some areas, these are constant, these are found in all the promoters, and this sequence within the promoter which is constant in all the promoter are known as the consensus sequence. And this consensus sequence is actually the main area which is identified by the RNA polymerase enzyme. Now come to the detail of prokaryotic promoter, that is the promoter of bacteria. So this is the transcription start point, and this side is the gene, and on the left side there is the upstream side. The promoter for bacteria RNA polymerase is located. So within the promoter of bacteria, there are two consensus sequences which is situated at, so if we count like this, so which is situated at 1 2 3 4 5 6 7 8 9 10. So we'll do it again, 1 2 3 4 5 6 7 8 9 10. So this is minus 10, and if we go on counting, so suppose this is minus 35. So there are two consensus sequences in prokaryotic promoter which are located at minus 10 position and minus 35 position. So these two consensus sequences, they have a specific sequence, and this sequence are same in all the promoters of different types of genes. So the sequence at the minus 10 position is T A T A A T T A T A A T. So it will start from minus 10 and minus 9, minus 8, minus 7, minus 6, minus 5, and minus 4. It will go up to, it will expand up to this location. So this sequence T A T A A T, here is 5 prime, and on this side it is 3 prime. This location, this sequence is known as minus 10 sequence or Pribnow box, after the name of a scientist who discovered it. So actually this is like this, A T A T T A. 5 10. This sequence is the minus 10 sequence, which starts from minus 10 position in the prokaryotic promoter. And there is one more such type of sequence which is called minus 35 sequence, and this sequence is T T Z A C A. So A C T Z T. So this sequence is known as minus 35 sequence. So these two sequence at minus 10 and minus 35 position, they are the consensus sequence for prokaryotic promoter. And these are called consensus sequence because if you see the promoter in bacteria, so this is promoter for a gene. So if you examine another promoter for another gene, then you will find that the other areas, the nucleotides which are present in these areas, these areas are not equal. But if you compare with another gene, then you will find in the next gene also the sequence at the minus 35 and the sequence at the minus 10 position are exactly similar, and that is why they are known as consensus sequence. And this consensus sequence, these sequences at minus 10 and minus 35 position is actually main responsible sequence which is identified by the enzyme RNA polymerase enzyme. And the other part, other than this consensus sequence, the other parts are not important for identification by the RNA polymerase enzyme. Now let us discuss about eukaryotic promoters. So before discussing about the eukaryotic promoters, we have to know that in eukaryotes there are 3 types of RNA polymerases. So they are known as RNA polymerase 1, 2 and 3. And So for each of these three types of RNA polymerase, there are three different types of promoter or promoter regions are found. So RNA polymerase 1 has their own promoter, 2 has their own, and 3 has their own promoter. So the RNA polymerase 1 is responsible for formation of mainly rRNA or the ribosomal RNA, and they form 28S rRNA, 18S rRNA, and 5.8S rRNA. The RNA polymerase 2 is responsible for formation of mRNA, while the RNA polymerase 3 is responsible for formation of tRNA, transfer RNA, as well as one ribosomal RNA, that is 5S rRNA. Today we are going to discuss about the promoter of RNA polymerase 2 only, and we are not discussing the promoters of RNA polymerase 1 and 3. Since we are dealing with the transcription, so that is why we are discussing the promoter of RNA polymerase 2, because the RNA polymerase 2, it forms mRNA, main transcription product. So the promoter of RNA polymerase 2 is a complicated promoter, and it starts exactly at the transcription start point. This is transcription start point, and there is a sequence at the transcription start point. It is situated towards left and right side, and this sequence is called initiator sequence, which is at the right at the transcription start point, and towards the upstream of the initiator sequence at the -25 position. At -25 position, there is a sequence which is called TA data box, and the sequence is TATAA. So this sequence is present at -25 position, and this consensus sequence is known as the data box. And there is one more consensus sequence which is located at -60 position, and this sequence is ZCCAATCT. Okay, so this sequence is present at -60 position, and this sequence is popularly known as cut box, CAAT, cut box. Okay, so there is one more sequence at -100 position. It is upstream of the transcription start point. This is called ZC box, guanine cytosine box, and the sequence is ZZC ZZZC ZZZC ZZ. Okay, so this sequence is present at -100 position towards the upstream of the transcription start point. So in the promoter of RNA polymerase 2, we found 4 elements are there. There are 4 regions which is important for binding of the enzyme to the promoter area, and the first is known as the initiator sequence, which is located right at the transcription start point, and next is at the -25 position, which is called the Tata box, and the next is the cut box at -60 position, and the fourth one is the ZC box, which is located at -100 position. One more important thing to know is that you see in this 2 strand of DNA, where this consensus sequences are read, they are actually this is 5 prime and 3 prime. They're read in the 5 to 3 direction of the DNA strand, which is actually the coding strand. Coding strand means the strand which is not copied into mRNA. So the opposite of the coding strand, that is this one, this one is actually copied as mRNA, and this strand is known as template. Okay, so this strand is template DNA, template strand. So while when we read the sequence in the consensus sequence, or when we read the sequence in promoter, then we read the sequence in the coding sequence only, not in the template sequence, and we read it in 5' to 3'. When we read the sequence in a consensus sequence of a promoter, then it is read from 5' to 3' direction of the coding strand. And what is coding strand? So coding strand is the strand which is opposite to the template. And what is template? So the template strand is the strand from which the mRNA or RNA is copied. So the when there is transcription, then during transcription, this strand will be copied as mRNA, and this strand will be known as template strand, and the opposite of this template is called the coding strand. Okay, so when we read the sequence in promoter, then we read or write only the sequence in the coding strand, not the opposite sequence which is in the template strand. Thank you. This is all about the promoter.